I'm actually going to begin my talk by just talking for a few minutes about my own experience fundraising for Dana-Farber, mostly through the Dana-Farber Marathon Challenge team, and Jan Ross is over there, director of DFMC, and there are other DFMC staff here that I want to give my little shout out to. My participation in the Dana-Farber Marathon Challenge was really the culmination of two separate sort of threads in my experience. One has to do with my devotion to Dana-Farber. So I first walked into the doors at Dana-Farber in 1980 when I was an intern, and I walked in and I saw what was going on there, and I said to myself, I need to work here. And in 1983 I was lucky enough to be able to come to Dana-Farber as a fellow, and I have voted with my feet by not moving my feet. I have stayed at Dana-Farber all of that time, and it's really because of the mission and the way this place is put together and the familial feel of Dana-Farber that's made me want to stay there, and I've always wanted to sort of give back. So there's that thread. Then the other thread is, I am so not an athlete. And my daughter will tell you, everybody will tell you that. But I've always sort of been fascinated by the Boston Marathon, and I thought, well, it'd be cool to run the marathon once. I didn't think I would ever be able to qualify, but then I heard about the Dana-Farber Marathon Challenge, and this was back in the mid-90s, maybe the 5th or 6th year that the DFMC had been in existence, and I thought, okay, I will get a charity number, it'll allow me to run, I get to raise money for Dana-Farber, and then I will be able to check the box. I've run a marathon, it's a bucket list kind of thing, and one and done, and it'll be great, and the Institute will benefit. The problem is that I met everybody on the team, and these are some of the most amazing people in the world. Their dedication, the things they want to do, the fun that they have, and I got totally swept up in the thing. I got seduced. I ended up running several marathons, and then when I couldn't do that anymore, I should say I came to running late, in my late 30s, early 40s, which is probably why I still have decent knees at age 62. But I was able to run a couple marathons, and then even when I stopped doing that, I joined the Dana-Farber Marathon Challenge board, and I've continued working with the board, and there's no way that I would walk away from that, and it's continued to be a major part of my life. So it's been great, and we've been able to raise a ton of money. Let me tell you about what some of that money is being used for. And the context in which I want to discuss this, the thing you have to know in order to understand from a research point of view how this money is being used, is that we are living through an absolutely unprecedented time in the history of cancer research. Actually, in the history of humanity, this is the first time we've been able to understand at a basic molecular level what really causes cancer. And this is incredibly important. This has huge impact. The impact is just as great as the discovery that microscopic organisms cause diseases like pneumonia or tuberculosis. Because the impact of that discovery was, now you could think about making antibiotics that killed those bacteria, and that cures the disease. And once you start thinking that way, you can take formerly incurable diseases like tuberculosis, a mystery for millennia, and actually cure it. And that's what's happening now with cancer. And the basis of that discovery is that cancer is a disease of genes. And let me explain that as simply as I can. Based on some discoveries by Dana-Farber scientists back in the 80s, we know that the process of cell growth, one cell becoming two, becoming four, and so on, that is not a passive process. That doesn't happen just because there's food and water around. Instead, it's a tightly regulated process that's controlled by genes. So we know there are about 25,000 human genes, and we think that maybe 1,000 of them are involved in regulating this process of cell growth. Now, genes don't do anything themselves. Genes are blueprints for the structure of proteins, and it's proteins that actually do the work in cells, and in this case, we're talking about proteins that regulate cell growth. It's a tightly regulated process. The cells in the skin of my finger are not growing now, so those cell growth proteins are turned off. But if I cut myself and my skin cells have to grow to fill in that cut, the protein gets turned on and tells cells to grow, and then when the cut is filled in, the protein gets turned off again and the cells stop growing. Now, the reason this is important to know is that we now understand in all cancers, essentially, the reason they occur is because there's been damage to these genes that are the blueprint for cell growth proteins. Damage, mutation, different structure, whatever it is, the blueprint has changed. So if the blueprint has changed, that means the structure of the protein has changed. And what often happens in cancer is that the structure of the protein is changed in such a way that it no longer toggles back and forth between being active and inactive. It's stuck in the active position, so it's constantly giving a signal for the cell to grow. And that is really what cancer is. But it provides us with a real opportunity. And this opportunity speaks directly to this notion of precision medicine that President Obama started talking about in his State of the Union this year. The idea of precision medicine is that we could come up with a drug that specifically targets that abnormal protein to turn off its effects. And when you do that, you get a decrease in the cancer. Sometimes you can make it go away with very few side effects. And the idea of precision medicine is that we will take this specific drug that is precisely targeted against the mutation in the patient's tumor who's sitting in front of me, the doctor, whoever the patient is seeing, and have these responses. The precision is about targeting the therapy to the patient and the patient's specific abnormalities. We know we're on the right track here because there may be 15 or so of these therapies that really do this, so-called targeted therapies, because they're targeted against the effects of these mutations. So it's a wonderful vision, it's a seductive vision, it's a compelling vision, but in 2015, we can't offer that for all cancer patients. So why not? There are really two major scientific obstacles to being able to offer this to all patients. The first is we don't know what all the mutations are in all cancers. We don't know what the landscape of mutations are. And that's one of the reasons that we began a project at Dana-Farber called Profile. The Profile project is designed to get a profile of the genetic abnormalities in the cancers and leukemias of all the patients who come to our hospital. It's a huge amount of data, but we take all those data, put them into a database, and make those data available for all of our researchers to go into the database and try to figure out what's the effect of a specific mutation on the behavior of a cancer in a patient. What's the effect of that mutation on the sensitivity or resistance of that cancer to the kinds of therapies we want to give? It's an amazingly powerful thing. We are now profiling somewhere between 5,000 and 6,000 patients a year. Year after year after year. This is going to be the largest database of its kind in the world. But nobody will pay for it. Government funding agencies won't pay for it. Insurers won't pay for it, even though we can sometimes find mutations in patients. You know, we can find the mutations that you really can direct drugs against, provide that information to providers like Eric, and they can give the right drug. And in fact, we've even been able to find mutations we didn't expect. That's the power of doing this for everybody. We find mutations we didn't expect in some patients who are quite honestly at death's door. We find mutations, treat them with the targeted therapy, and they walk out of the hospital. We're calling these our Lazarus cases. But nobody will pay for this. The only way we can get this done right now is through philanthropy. And it's through fundraising efforts like yours that we can get all this information to understand how mutations drive cancer and these Lazarus cases as well. So the first reason we can't provide this precision medicine to everybody is because we don't know the landscape of mutations. The second reason, which in some ways is even more important, we don't have drugs yet. We don't have drugs to fight the mutations we know about, much less the ones that are remaining to be discovered, the new ones. Now, ordinarily, we partner with drug companies to try to come up with these new agents. And for a variety of reasons we can talk about, our partners have not been so great about that. But we can't abandon our patients. So at Dana-Farber, we have hired organic chemists who synthesize molecules in their laboratories to target these mutations. We have 5 of them now. 3 of our chemists have come up with drugs that are actually in clinical trials. But again, nobody will pay for this. Government won't pay for it, foundations won't pay for it, companies won't pay for it. It's all too risky. The only way we've begun a program in what we call chemical biology is through fundraising. So what I'm trying to tell you is that these incredibly important, cutting-edge, high-impact studies that really will impact patients depend entirely on the kind of work that you do. So if you're looking for some way that the work that you do impacts patients and researchers, you need look no farther than the faculty who work at Dana-Farber. So all of you who run, ride, walk, swim, play golf, we just thank you from the bottom of our hearts, and you can also know that patients now and in the future will thank you as well. So that's my